Hello and welcome. Today, we're diving into the fascinating world of engineering biology, sometimes called synthetic biology or synbio. What is it? Imagine combining biology with engineering, computer science, and chemistry. Instead of just studying life, we're actively designing and building new biological parts, devices, and systems, or even redesigning existing ones for useful tasks. It's about shifting from simply observing biology to engineering it. So how is this different from traditional biology? While traditional methods focus on understanding what's already there, engineering biology applies engineering thinking. Think standardization, creating reliable biological parts, like Lego bricks called biobricks, that anyone can use. Think abstraction, focusing on the big picture without getting lost in every tiny detail. Modularity is key too, designing components that can be easily swapped and combined. And crucially, it relies on a design-build-test-learn cycle. You design something, build it in the lab, test if it works, learn from the outcome, and then refine the design. It's an iterative process, much like developing software or machines. What tools make this possible? First, DNA synthesis lets us literally write DNA code from scratch, creating new genes. Then, DNA sequencing allows us to read that code quickly and cheaply, verifying our creations. Gene editing tools, especially CRISPR-Cas9, give us incredible precision to modify existing DNA. Metabolic engineering allows us to rewire an organism's internal chemical factories to produce specific substances. And sophisticated computational modeling helps predict how our designs might behave before we even step into the lab. The potential applications are staggering. In medicine, think microbes engineered to produce drugs like insulin, smart biosensors for diagnostics, engineered immune cells fighting cancer, like CAR-T therapy, and faster vaccine development. For materials, imagine bacteria making sustainable plastics or self-healing fabrics. In energy, microbes could produce biofuels or hydrogen gas. Environmentally, we could design organisms to clean up pollution or capture carbon dioxide. Agriculture could see crops enhanced for yield, nutrition, or resistance to pests and drought, potentially even fixing their own nitrogen to reduce fertilizer needs. Even our food is changing, with developments in cultured meat and engineered yeasts creating new flavors and ingredients. Of course, it's not without hurdles. Biological systems are incredibly complex and sometimes unpredictable, making design challenging. Scaling up lab experiments to industrial levels isn't always easy. There are also important safety concerns about accidentally releasing engineered organisms and security concerns about potential misuse. And inevitably, profound ethical questions arise. We need careful discussions about creating synthetic life, modifying genomes, and ensuring everyone benefits fairly from these powerful technologies. Engineering biology is moving fast. It offers truly transformative potential for tackling global challenges in health, sustainability, and more. By applying engineering principles to life itself, biology is becoming programmable, almost like software. What was once science fiction is becoming reality. But as we explore this exciting frontier, responsible innovation and open dialogue about the ethics involved are absolutely essential. Thanks for tuning in.